All right, so today's lecture is going to be on the endocrine system. My name is Chris. I'm one of the MCAT tutors here, and this is one of the lectures, part of our 15-week program. So just a little bit of background information here. I put this on all of my slides. I put a couple of links up where you could get in touch with the group, get in touch with some other students that are a part of the group. So we have the group me over here. The Instagram handle is @SocraticMed_. Our YouTube link to our channel is over here. You can find all of our videos, segmented videos, and all of our video lectures uploaded onto the YouTube channel. And the course, we do 3 lectures a week on Thursday, Friday, and Saturday at 8:30 p.m. Eastern time. And every Sunday, we hold unstructured office hours that start at 8:30. So you can come to those if you have any questions. Okay. Here it is. So I just wanted to look at this quickly as well. So these lectures are going to be recorded and put on YouTube. So I just wanted to show this consent to audio or video recording release. So by continuing on this lecture, you're agreeing to this release. And if you want to change your name, maybe get rid of your last name or put an initial or get rid of your profile picture, if that makes you more comfortable to be on the lecture, then now would be the time to do so because this video will be uploaded to YouTube. And the way I do lectures is just that you keep your mics muted. And if you have any questions, you can use the chat box, or I'll give you a chance to unmute at the end of the section before we do questions. Janelle is another one of the tutors, and she's on here today. So she's also going to be helping me out with the chat box, answering questions. Okay, so we're going to start just with a brief overview of the endocrine system, which is the system that controls hormones in your body. So what is the endocrine system? So the control system of the body that is slow acting and long lasting when compared with the nervous system. So the nervous system, if you remember from the past lectures, is a very quick acting system. It uses electrical impulses. It can transmit information throughout your body, from one end to the other in a fraction of a second. The endocrine system, in contrast, is slower acting and it's more long lasting. So it has a longer lasting effect on the target tissue that it reaches. So like I said, while the nervous system signaling is usually mediated by electrical impulses carried by neurons, endocrine signaling is carried out by hormones released into the blood and circulated to other tissues. So that released into the blood is a very important part of the definition of what an endocrine molecule is. Hormones are released into blood. There's another term that we'll talk about in a little bit called exocrine, and that means that it's released into a duct that's not actually released into the blood. But anytime you hear endocrine or hormone, by definition, they have to be released into the blood to circulate to other tissues. So endocrine signaling can be preceded by nervous signaling, and it usually is. And we're going to talk about the HPA axis later. That's basically how your body reacts to stress and how it bridges your nervous system and your endocrine system to create a holistic response. So the endocrine system uses a variety of types of signaling and a variety of hormones to affect the body, obviously. So there are a bunch of different hormones and there are a bunch of different receptors on all of the target tissues. But there are actually different types of signaling that the endocrine system can use. So I put those on this slide over here. So we're just going to go over them. So there are 4 types of endocrine signaling that you should be able to identify for the MCAT. So you definitely want to be familiar with all of these, at least the first 3. So the first one is just straight endocrine signaling. So the target tissue is not in close proximity to the endocrine tissue. An example, insulin is produced in the islets of Langerhans in the pancreas, and it circulates throughout the body to interact with distant muscle tissues. So that's what straight endocrine signaling is. That's basically just the definition of what the endocrine system does. The target tissue is not close to the specific cell. It releases hormones, enters the bloodstream, and it can circulate to a farther distance and interact and affect change in that tissue. The second type is paracrine signaling. So the target tissue is in very close proximity to the endocrine tissue. So it's endocrine signaling, but it's just closer. These paracrine factors are released directly into the extracellular matrix instead of the bloodstream. So an example of paracrine signaling would be the release of neurotransmitters into the synaptic cleft. So they're not touching each other and they're not communicating with each other via electrical impulses. They're communicating with each other through this release of signals. So, and because they're so close to each other, it's considered paracrine signaling, because that's how one molecule is relaying information to the next, one cell, excuse me. The third type is autocrine signaling. So the endocrine tissue also expresses the receptor for its hormone. So the cell that creates the actual hormone is also expressing the receptor and is also receiving the hormone. So you could kind of see how that becomes sort of positive feedback loop, and it could be, it's okay if it's in a controlled environment, and it could also be problematic if it's not controlled. So this type of signaling is often found during growth and development, like embryonic development. I think early childhood development, there's autocrine signaling in certain types of cells. The specific types of cells are not too important. It's just, you should know that autocrine signaling is really mostly found in development and in your immune system. So during T cell activation, the T cells will actually release factors that it actually expresses a receptor for. So it results in what we call monoclonal proliferation, which is, it just makes a bunch of clones of itself, like I said, in that sort of positive feedback loop. So this type of signaling is also often found as a pathway for cancer growth. If you've ever taken a cancer bio class or you've ever studied cancer, you'll see that a lot of cells, one of the ways in which they evade these natural checkpoints and the ways which they continue to grow despite what the body thinks is natural, is that they take advantage of this autocrine signaling pathway. And that could lead to tumor growth and eventually metastasis of the cancer. So the fourth type of signaling is not one that I've seen very often in my own studies, but I did see it a couple of times upon revisiting when I was making the lecture. So I thought it was at least a good idea to include it. And there's not a lot of these types of signaling, so I think it's probably a good idea that you guys do know it for the MCAT. So it's called juxtacrine signaling. It's also known as contact-dependent signaling. So it depends on the type of connection between two adjacent cells. So it seems kind of similar to paracrine signaling, but juxtacrine signaling, the cells are actually touching each other. So there are three different types. Juxtacrine signaling can be broken down into three types of signaling, and I put it right under that. So the first one is cell-to-cell signaling, and the ligand of one cell's receptor is found on the membrane of an adjacent cell. So one cell, the membrane is expressing a protein that's maybe sticking out of the membrane, and that protein actually hits the receptor of another cell, and that's considered juxtacrine signaling. Gap junction signaling, two adjacent cells communicate by a direct connection. So think back to electrical synapses when we were talking about the nervous system. That's a form of gap junction signaling, because these two cells are connected between these gap junctions, and they can communicate with each other that way. And then the third one is cell extracellular matrix signaling. A cell's receptor is activated by glycoproteins found on the extracellular matrix. So it's similar cell to cell, but it's just cell to extracellular matrix. So there's some sort of protein or glycoprotein that's in the extracellular matrix that is interacting with the receptor on a specific cell, interacting with a specific receptor on the cell membrane. And this is one of the ways that cells like interact with each other. And just spread communication with each other. And it's important for like cell proliferation and making sure that none of that goes out of check. Okay, so now we're going to talk a little bit about the different types of hormones. Hormones are generally derived from three different types of precursor molecules. So the first one I want to talk about are lipid-derived hormones, and in humans, it's primarily the steroids. Most of these hormones are derived from cholesterol and are lipid-soluble, which means they're hydrophobic. Sometimes they'll say hydrophobic, sometimes they'll say lipophilic. It all means the same thing. They're just non-polar molecules. And if you look at most of these molecules, they have very similar structures to cholesterol. So if you know what cholesterol looks like and they give you the structure of a hormone and they're asking you to identify it, it might be pretty useful to know the structure of cholesterol so you can easily identify them. And lipid-derived hormones can diffuse through the cell's phospholipid membrane to act on interior receptors. So remember, they are hydrophobic. and so is the phospholipid membrane. So these cells are able to diffuse straight through without any sort of transport protein or anything like that, and they act inside. So since they're not water soluble, like we just said, steroids rely on transport proteins to get through the blood. So albumin is one of the bigger transport proteins that you find in the body. And they usually will just like via nonpolar interactions, they'll just stick to albumin and they'll circulate the body. And then once they get to the target tissue, they will dissociate from albumin, go through the membrane, and they'll enter the cytoplasm. So steroids often affect the body slowly since they directly affect RNA transcription. So a lot of steroids act directly on RNA transcription, and that's their function. It's not like a peptide hormone, like we'll talk a little bit how it alters the enzymatic, like the enzymatic stuff that's going on inside the cell. These actually directly affect RNA transcription and what is being transcribed. So because of that, their effects can persist for weeks in the body. And they're synthesized in the Golgi on demand, and they're shipped immediately since they can't be stored in membrane-bound vesicles. So the vesicles in your cell, they're very similar to the cell membrane. They're hydrophobic. They're usually made of phospholipids. So we just said that steroids can't, they're hydrophobic, so they go through these membranes. So we can't synthesize steroids and store them because they'll just leak out. So that's why we actually make them on demand. They're synthesized in the Golgi and they're immediately shipped out. They're very short lasting and they affect their target cells very quickly. And a little bit about the nomenclature. Steroids will often end with the ONE or OL suffix. So we have estradiol, which is an estrogen, testosterone, aldosterone, and cortisol. Those are all lipid-derived hormones. They're all steroids that we find in the human body. And we'll talk a little bit about specific hormones later in the lecture. Okay, so the second type of hormone that I want to talk about are peptide hormones, which are probably the ones that we're most familiar with because we talk about a lot of them. So these hormones are derived from peptide precursors and they're produced in the rough ER, obviously because the rough ER is where where most of the cell's ribosomes are. So the peptide hormones are mostly produced in the rough ER. So peptide hormones, unlike steroids, are hydrophilic. They are polar and they can dissolve in water. Therefore, they cannot diffuse through membranes like steroids can. So also, since they can't diffuse through membranes, our cells are able to store them in vesicles. So these types of hormones are often produced in advance and they're stored until they need to be utilized. So make sure you understand that conceptually, that peptide hormones, we can create them in advance and we can store them. So we can release them pretty quickly once we need them. The steroid hormones we have to produce on demand. So it takes a little bit longer because we have to produce them, but we immediately ship them out because we can't store them. So peptide hormones, since they are hydrophilic, they can dissolve in the blood plasma. They don't need transport proteins because they are proteins. And they are transported to receptors on the surface of the cells, on the surface of the target cell's membrane. So they don't diffuse through. The receptors are on the cell's membrane on the outside, sticking out. So peptide hormones bind to a receptor. The hormone initiates a signal cascade inside the cell, and that alters the existing enzyme activity in the cytoplasm. The effect of the peptide hormones are relatively quick, and they begin minutes to hours after secretion. And peptide hormones often, but not always, end in IN. So that's a pretty common protein ending. So like insulin, for example, is a peptide hormone. Oxytocin is a peptide hormone. But then again, follicle stimulating hormone is also a peptide hormone, and it doesn't end in IN. So if you see IN on the exam and they're talking about a hormone, it's safe to say that it is a peptide hormone. But the other ones, just because it doesn't have an IN doesn't necessarily mean it's not a peptide hormone. And FSH is a pretty common one that you'll see on the MCAT. So you should commit to memory the fact that it is a peptide hormone. And the third type that we're going to talk about are amino acid-derived hormones. And those are the hormones that are derived from single amino acids, whereas peptide derivatives are created from polypeptide precursors. So there are multiple amino acids that are hooked up, and those are the ones that create peptide derivatives, but the amino acid hormones are just, they use just singular amino acid precursors. And since the hormones are so small, they can take on properties of the other two groups of hormones depending on where they're synthesized. So some amino acid hormones will act like peptides, and some of them will act like steroids. So the catecholamines, epinephrine, for example, these are the hormones that are secreted by your adrenal glands. The catecholamines don't go through your cell membrane. They actually bind to cell surface receptors, and they're pretty quick acting. So in that way, they're very similar to peptide hormones. T3 and T4 are two of your hormones that are secreted by the thyroid. They have an iodine component, and they're actually able to diffuse into the cell, and they act on interior receptors, a lot like steroids and lipid-derived hormones that we were just talking about. So both of these catecholamines and the thyroid hormones, they're both amino acid-derived hormones, but you can see they act very differently. Their behaviors actually resemble those of the other two categories. But for our categorization purposes, we divide them into three based on their precursors. So that's a pretty important thing to know about the amino acid-derived hormones. And before we go on to this question, does anybody have any questions about what we just went over? You can use the chat box, you can unmute. And if not, I will give you guys a minute or two to look over this question. Remember, the polls are anonymous, so give it your best bet. Give me 1 second. Hold on. All right, sorry about that. All right, so the poll should be out. Take a minute. And there's the question. All right, take another couple of seconds, and then I'm going to close the poll. All right, so 100% of you said C. And the answer that I had was B. So let's go over it. A student is studying an unknown hormone. The hormone is observed attaching to proteins in an aqueous environment. If the student wanted to find the receptor on which this hormone acts, she should look. So A, B, C. A is in the blood. I wouldn't really look for any hormone receptors in the blood. I mean, unless you specifically learned about something that acts on blood tissue, but we haven't really been talking about that in this lecture, so I'm glad nobody picked that one. D, I also would not say, because I just don't think we would find a receptor in the extracellular matrix. I mean, there are, the extracellular matrix has, like, when we talk about juxtacrine signaling, it can act on a cell membrane, but it doesn't really have receptors. So B and C, I guess, were the two top choices. The reason why I would say it's B is because steroid hormones attach to proteins in an aqueous environment. So those are the lipid-derived proteins, and those are the ones that we said attach to albumin in the bloodstream. So the aqueous environment that you find hormones in.
Really hard to stay focused on.
Yeah, so we would most likely observe that this is a lipid-derived hormone, and go through the membrane, and they act on internal receptors. If it were on the cell membrane, it would be a peptide hormone, and since peptide hormones are hydrophilic, they don't actually attach to proteins in the aqueous environment. They just dissolve by themselves. So that's why it wouldn't be C, because those peptide hormones are the ones that act on the cell membrane. The steroids are the ones that attach to proteins, and they actually diffuse into the cell to act on those internal receptors. Is that clear for everyone? Okay. So you said I was thinking it wouldn't get through the membrane like steroid hormones, therefore it would need a receptor. I could see how that seems kind of tricky, but I think the main thing to keep in mind is that if it's attaching to a protein in the environment, it's going to be insoluble, and it's going to be a lipid-derived protein. That's really the key part of the question. And any hormone that attaches to the protein is going to diffuse through the cell, membrane. Exactly. Yeah. The protein would be albumin, is like a protein that you should probably be thinking of when you see this question. Yeah. Cool. Okay, so the next part of the lecture is going to be about portal systems and the HPA axis. I put a little graphic at the bottom. It just kind of demonstrates very basically what the negative feedback loop regarding this specific hormonal pathway is, and we're going to talk about that a little bit moving forward. So a little bit about portal systems. They are small circulatory systems consisting of two capillary beds in very close proximity, and they facilitate the quick release and transport to target tissues of hormones. So normally in circulatory pathways, they only go through one capillary bed before returning to the heart. And that's because when blood goes through a capillary bed, there's a significant drop in hydrostatic pressure. So it really needs to make it to the heart again in order to reoxygenate and re-up on hydrostatic pressure, because otherwise it's not really going to be able to get any nutrients into the surrounding cells. Fortunately, with this portal system, the two capillary beds are very close to each other. And it just ends up working that after the blood goes through that first capillary bed, it ends up going through the second one, and it can release all of the hormones in that second capillary bed, and it doesn't actually get into systemic circulation. So the first capillary bed, like I just mentioned very briefly, allows the hypothalamus to release releasing or inhibiting factors that will travel an extremely short distance before reaching the next portal bed that connects the next capillary bed that connects to the anterior pituitary gland. So in that first bed, the first capillary bed is by the hypothalamus, the second capillary bed is by the pituitary gland. So in the first one, these tropic hormones, which we're referring to as releasing or inhibiting factors, they enter that first capillary bed, They travel a very short distance, and then they reach the next capillary bed where they actually diffuse into the pituitary gland. So this second capillary bed actually allows the hypothalamic hormones to be taken up by the pituitary gland without getting into the systemic circulation. So humans have two portal systems. We have this portal system. It's called the hypophyseal portal system. I think I have it on one of those slides. And the other one is the hepatic portal system with your liver. There are actually two portal beds that connect your digestive system and your liver, because your liver actually needs to filter some toxins out of what you eat before it reaches your heart and before it enters your systemic circulation. So the hypothalamus, the first stop in the HPA axis. The hypothalamus has multiple roles in daily functioning. We may have spoken about it before in previous lectures when we were talking about the brain. It controls circadian rhythm, controls body temperature, hunger, it manages stress levels, and it houses the brain's reward center, the dopamine-driven reward center. But we're not going to be talking too much about that in today's lecture. We're just going to be talking about its hormonal control on the body. So the hypothalamus exerts endocrine control over the body in two ways. So the first way, which we've previously discussed, is by sending releasing or inhibiting factors through the portal system to the anterior pituitary lobe to control its production of other hormones. So the pituitary lobe is split, the pituitary gland is split into two lobes, and we're going to talk a little bit more about that on the next slide. And the hormones that are released by the hypothalamus include corticotropin-releasing hormone, growth hormone-releasing hormone, and gonadotropin-releasing hormone, and thyrotropin-releasing hormone, among others. So you see this tropin-releasing hormone. These are the releasing factors. These are the tropic hormones that we're talking about. Tropic hormones are any hormones that control the inhibition or release of another hormone. So the hypothalamus and the pituitary both release these tropic hormones. And the hypothalamus also creates its own hormones and delivers them to the posterior pituitary gland where they are released. So the hypothalamus releases tropic hormones that allow the anterior pituitary to release its own hormones, but it also creates hormones and it delivers them to the posterior pituitary. So I'm going to clarify this on the next slide. So if it's confusing, don't worry. We are going to talk about it right now. So the pituitary gland, if you look on the left, it's a tiny, tiny gland that sits right at the base of the middle brain. And it is a small gland. It maintains contact with the hypothalamus through the hypophyseal portal system, the anterior lobe, and the pituitary stalk, which is how the posterior lobe is connected to the hypothalamus. So the anterior lobe is also known as the adenohypophysis of the pituitary gland, is made up of true endocrine tissue. So when we say true endocrine tissue, we mean that it's actually producing the hormone that it's secreting. So the anterior lobe is true endocrine tissue because it produces and secretes the hormone. So upon receiving hormonal input from the hypothalamus via that portal system that we just discussed, the anterior lobe will produce and release up to 7 hormones. So this is follicle stimulating hormone, luteinizing hormone, ACTH, I think it's adrenocorticotropic hormone, TSH is thyroid stimulating hormone, prolactin, endorphins, and growth hormone are all released by the anterior lobe of the pituitary. So I've seen a lot of different study materials. They use the acronym FLAT-PEG, and I think it's pretty helpful because it's the one that I use and it's the one I used for the MCAT. So FLAT-PEG is the acronym that will help you remember the anterior lobe, what is created in the anterior lobe. So FLATPEG. You definitely want to know those. I do see them pop up every once in a while, and even if they pop up on like the psychosocial in some sort of passage, it's always a good idea to have background information of it. So that's the anterior lobe. And we also have the posterior lobe, which is known as the neurohypophysis. And the posterior lobe, in contrast to the anterior lobe, is not made of true endocrine tissue. So that means it's releasing hormones, but it's not creating the hormones. So we don't consider it a true endocrine tissue. It releases hormones that are produced by the pituitary, that are produced by the hypothalamus. So we talked before about the, right over here, hold on. I thought it was on that slide. The pituitary stalk is what I was looking for. That's how the posterior lobe is connected to the pituitary gland. Oh, it's down here. Okay, the hypothalamus produces oxytocin and ADH, which is antidiuretic hormone, if you remember. So it travels down the pituitary stalk, which is just an actual structure of the brain that connects the hypothalamus to the posterior lobe of the pituitary stalk. And from the posterior lobe, it is released into the body. So the way I remembered it, because I kind of had anterior and posterior lobes confused in my mind. So to remember which lobe does not produce its own hormone, you can remember that the posterior lobe is an imposter endocrine gland. That helped me a lot because I always confuse the two. So if you can remember that the posterior is the imposter and you remember the two hormones that it secretes, which are oxytocin and ADH, which is vasopressin, ABP, arginine, vasopressin. That's all the same hormone. I would recommend using that trick. Okay, and we're going to talk about the third stop on the HPA axis, which are the adrenal glands. So while the pituitary gland has multiple other target organs, the adrenal glands are the final stop in the HPA axis. So the adrenal glands receive input from the pituitary gland to secrete hormones that regulate metabolism, immunity, blood pressure, and the response to stress. So each adrenal gland is composed of two sections. We have two adrenal glands. They sit on top of each of your kidneys. If you look over here at this graphic, the top right of the graphic, you can see the adrenal glands sitting right up top. The prefix ad means on top, renal means kidney. So we have two of these glands sitting on either kidney. And the adrenal gland is composed of two different sections. The adrenal cortex, which if you look over here is everything, all of these layers except this middlemost part. That's called the cortex, because remember, anytime you see the word cortex, they are referring to the outer part of whichever structure we're talking about. And the adrenal cortex produces a lot of different types of hormones. It's the glucocorticoids, which cortisol is one of the more popular ones. Mineralocorticoids like aldosterone. Remember, aldosterone helps us intake sodium and ions so that we can take up water and increase our blood volume and blood pressure. And androgenic steroids like testosterone are also created in the cortex in smaller amounts. Most of the testosterone is produced in the cells of Leydig in the testes, but the adrenal cortex does also produce a small amount of testosterone. So the adrenal medulla is the innermost part. Just like if you think of the renal medulla, it's the innermost part. If you think of the medulla oblongata in the brain, it's inside the brain. And it mostly produces the catecholamines like epinephrine, norepinephrine, dopamine. Those all come from inside the adrenal medulla. Okay, and then just a summary of the hypothalamic pituitary adrenal axis. So while your endocrine system involves many other organs, some of which we will discuss later in further detail, the HPA plays an essential role in the body's response to stress and its regulation of homeostasis. So negative feedback is super important in homeostasis. That's how a bunch of systems in our body maintain homeostasis and how they keep in check. The HPA axis is regulated by negative feedback. For example, high cortisol levels will stop the secretion of tropic hormones from both the hypothalamus and the pituitary gland. So if you look to the left, the graphic, we start at the hypothalamus. It releases CRH. It goes through that portal system and it interacts with the anterior pituitary. When the anterior pituitary receives that CRH, it relieves ACTH. The ACTH travels through the bloodstream and it touches the adrenal glands. It acts upon the adrenal glands and that releases cortisol. So cortisol will also act on other target cells. It'll go to your brain. It is like the stress hormone. It makes you feel stressed out. It increases, it mobilizes your glycogen storages. So it's part of your fight or flight response, the sympathetic response. But your body always needs a way to come down after one of these responses because any sort of stress that you're exposed to is not permanent. It's just a temporary stressor. So as cortisol levels rise in the blood, it feeds back to both the hypothalamus and the anterior pituitary. So it acts on both. And when it acts on both of these structures, it actually decreases the secretion of CRH and ACTH. So that in turn will decrease the levels of cortisol. And that's basically how negative feedback works. And we see negative feedback in almost all of the bodily systems that we study that we're going to see on the MCAT. So it's a very important thing to know conceptually. And before we do this, does anybody have any questions about what we just discussed? Okay. Okay, so I'm going to relaunch the poll. I'll give you guys a little bit. Take a look, and we'll regroup and discuss in a minute. All right, get your answers in. I'll give you like another 15 seconds. Remember, these are anonymous, so just give it your best shot and we'll go over it. All right. Okay, so 50% said C, 50% said D. And the correct answer is C. So let's take a look. When experiencing an increased blood concentration level of cortisol, all of the following would happen except. So that except, we're looking for what wouldn't happen when cortisol levels increase. So an eventual decrease in ACTH secretion, that would happen when cortisol levels increase in the blood, because remember, the negative feedback loop that cortisol is involved with, it feeds back and it actually inhibits the pituitary gland from secreting ACTH the higher the cortisol levels get. So if we see an increase in blood concentration of cortisol, you will see an eventual decrease in ACTH. And you're also going to see a decrease in CRH for the same exact logic. So it acts on the pituitary to decrease ACTH secretion. It also acts on the hypothalamus to decrease CRH secretion. You would also see an increase in gluconeogenesis, because that's just another function of cortisol. It mobilizes our glucose storage, and it provides energy. So the only thing that wouldn't happen would be C. So immediate feelings of calm and relaxation are not something that coincides with the release of cortisol. Cortisol is the stress hormone. So you would probably be feeling more stressed out or more anxious. And it would probably coincide with like an activation of the sympathetic nervous system. So you would not feel calm or relaxed with it. So that's why C is the correct answer. Does anybody need clarification on that? I can definitely explain if you do. Okay. All right, if you think of any questions along the way, again, feel free to put them in the chat box. Janelle can answer them, and if I see them in time, I'll also try to answer them. Okay, so the other organs of the endocrine system, we're going to talk about a few more, some of the important ones that are pretty high yield on the MCAT. And one of the more important ones is the thyroid gland. So if you look at the left, it's that sort of symmetric gland that sits in your neck. So the thyroid is a two-lobed endocrine gland that sits in the center of the neck below the Adam's apple in humans. And its main function is to regulate metabolism and balance calcium levels in the blood. So we mostly know it for its metabolism regulation. I'm sure you guys have come across like hypothyroidism and its effects. The thyroid is something I think that the MCAT does like to test. I've seen a lot about hyper and hypothyroidism on it, and it'll ask you, like, you have to have knowledge of how the thyroid gland works into the hormonal and endocrine system in order to answer the questions. So its main function, said that it regulates metabolism by producing two hormones in response to TSH released by the pituitary gland. So remember flat peg, the T in flat peg is TSH, thyroid stimulating hormone. Thyroid stimulating hormone is a tropic hormone because it is acting on another endocrine gland to release different hormones. So it's a releasing factor, basically. And the two hormones that are released by the thyroid are T3 and T4, which is what they're more commonly known as. T3 is triiodothyronine and T4 is thyroxine. So both of them basically do the same thing. They're just slightly different. I think T3 has one less iodine than T4, but they both increase the basal metabolic rate when they're released into circulation. So I think one of them is faster acting. I think T4 might be faster acting. But they both do the same thing. It's not super important that you strike a difference between the two. They're both released by the thyroid and they increase basal metabolic rate. But on top of the thyroid's role in regulating basal metabolic rate, it also has specialized C cells that release calcitonin. So calcitonin counteracts the effects of parathyroid hormone, which we'll talk about in a minute, and it decreases blood calcium level. So calcitonin decreases blood calcium level. It can increase calcium storages on your bone. It can act on your intestines to decrease calcium reuptake. It can act on your kidneys also to decrease calcium reuptake. But that is what calcitonin does. It decreases your calcium levels in the blood, and parathyroid hormone does the opposite. So again, I don't really have a great mnemonic for remembering those two. You kind of just have to remember them. I still sometimes get the two of them confused. But you definitely want to keep them straight for the MCAT. So what we were just talking about, the parathyroid hormone, parathyroid glands, excuse me. Humans have 4 rice-sized parathyroid hormones. Parathyroid glands situated behind the thyroid gland. So if you look at this graphic to the right, it shows the thyroid gland like we saw in the last one, and you see these 4 little dots that are sort of sitting 2 on either lobe of the thyroid gland. Those are the parathyroid glands. So they're very tiny, but it's because they only have one function, basically. So the thyroid secretes calcitonin to reduce blood calcium levels. The parathyroid gland secretes parathyroid hormones to increase blood calcium levels. So they sort of work in tandem to adjust your calcium level because it's a pretty important ion. It's involved in a lot of nervous signaling and a lot of muscular contraction. It's important to note that this is the only function of the parathyroid hormone. So as appropriate calcium levels in the bone and blood are vital for normal bodily functioning. Right. So it mitigates blood calcium levels, but it's also important to look at bone calcium levels. When bone calcium levels drop too low, that's how osteoporosis develops. The bones can get brittle, they can break. So these two are pretty important. And like I said before, I think the MCAT pretty often does like to test sort of deficiencies in this sort of calcium system. And next we are going to talk about the pancreas. So the pancreas is involved in digestion as well as the body's endocrine functioning. So you'll see it come up in the digestion lectures, and you'll also see it come up in lectures about hormones in the endocrine system. During digestion, the pancreas secretes digestive enzymes that are released through a duct into the intestines. So I'm sure you've learned about those enzymes. We're not going to go too much into them because since they are released into a duct and they're not released into the blood, they're considered exocrine enzymes. It's an exocrine function, so it's not technically considered a true endocrine function. So it's a little bit outside of the scope of this specific lecture, but we do have a digestion lecture that's probably going to touch upon a lot of the stuff that I'm about to talk about. I think it's next week, but this is a great, this and the next few slides that I'm going to do are a great, it's great groundwork for you to understand the digestive lecture a little bit better from an endocrine perspective. The pancreas also has tiny clumps of specialized endocrine cells known as the islets of Langerhans, which we mentioned a little bit earlier. So these cells produce insulin and glucagon, among a few others. I think it might also produce somatostatin. But insulin and glucagon are really the two that I see the most on the MCAT. They're both peptide hormones, like we were talking about before. So remember, they are water soluble. They readily dissolve into blood plasma, and they affect receptors that are on the cell membrane. They don't diffuse through because they are not lipophilic. They are hydrophilic. So this is an example of an endocrine organ that is not directly controlled by the pituitary gland. I just wanted to throw that in there just to contrast it from the HPA axis, because the pituitary gland does have a lot of endocrine control over the body. But insulin and glucagon are controlled by a bunch of other factors. It's time of day, your circadian rhythm, the food that you intake in your stomach. Those are all factors that affect insulin and glucagon secretion. Yep, cool. Janelle confirmed. So insulin, glucagon, and somatostatin. And I'm pretty sure somatostatin is, yeah, I think they come from the delta cells in the islets of Langerhans. Okay, so another, the ovaries and testes are another target cell and a point of interest when we're studying the endocrine system. So the pituitary gland can also release hormones that directly affect sexual functioning in both males and females. So follicle stimulating hormone, like we talked about in Janelle's previous reproduction lecture toward the beginning of the program, they help the maturation of primary oocytes into mature ova in females. And it is also present in males too. It helps the production of sperm, which takes place primarily in Sertoli cells. Luteinizing hormone helps the production of estrogen in females, and it encourages ovulation in females. So like we spoke about, FSH and LH are huge players in ovulation in females. Luteinizing hormone is sort of that last interaction that actually allows ovulation in females. The egg comes out of the follicle. In males, luteinizing hormone stimulates the production of testosterone from the cells of Leydig. And along with producing gametes, the ovaries and testes produce sex hormones that contribute to the body's normal metabolism. So they do produce gametes. We have ova and sperm that are produced. We know that that is basically their primary function, but they're also super important in regulating sex hormones throughout the body. So they contribute just to the body's normal metabolism, and they also help develop secondary sex characteristics during development, specifically during puberty. So does anybody have any questions about what we just went over before I launch this next poll? All right. You can put them in the chat if you want. I'm going to start the poll. Take a minute or so. Remember, this is anonymous, and we're going to regroup and discuss in a minute. Okay, polls are open. All right, take another few seconds if you have to get your answers in. Okay, I'm going to end the polls. Cool. So, oop. All right, so all of you said D. An increase in the anterior pituitary lobe function would most probably directly increase blood concentration levels of which of the following? Awesome job. That is correct. TSH is thyroid stimulating hormone, and that is one of the 7 flat peg. So if pituitary lobe function is going to increase, we would probably expect to see, out of these 4 at least, we would expect to see an increase in TSH. So great job with that. Vasopressin, remember, is also known as AVP, arginine vasopressin, antidiuretic hormone, ADH, and that is created by the hypothalamus and it's secreted by the posterior lobe. So anterior pituitary lobe function would not affect it. And same thing with oxytocin, and insulin comes from the pancreas like we spoke about before. There are a lot of other factors that are affecting it. So yeah, D is correct. You guys all got it. Awesome job. So I'm assuming no clarification is needed, but if you do, please type in the chat box and we'll talk about it. But I'm going to move on. Okay, so regulation of endocrine function and review of hormones. So here's the negative feedback that I put over here again, just because it's a pretty important concept. You have the stimulus. It acts on the sensor. The sensor is whatever structure is taking in input from the stimulus. It brings it to the control. The control is whatever structure is comparing it to a normal. So say your body gets really hot, the thermoreceptors in your body would be the sensor, and then it would go to the hypothalamus, which would be the control, because it's going to compare it to your regular body temperature. So if it sees that these thermoreceptors are receiving too much input and that your body is too hot, it's going to go to the effector. So the effector in this case would be maybe increased ventilation rate or an increase in sweating, because that's what's going to mitigate the effect of the stimulus. And then it feeds back negatively to the stimulus, and it slows down the entire process. Okay, and another diagram. So as we mentioned earlier, most of the endocrine system is regulated by negative feedback, although positive feedback we do see sometimes. In other words, the product or result of the signal cascade will directly inhibit the specific signal cascade upon increasing concentration and will activate it upon decreasing concentration. So this graphic, calcitonin, for example, is secreted by the C cells in the thyroid when blood calcium levels are too high. The increase in calcitonin will drive calcium levels in the blood down. When calcium levels are low enough and they're too low, the thyroid is going to stop producing calcitonin. And it's just an ongoing cycle of repeating. So I didn't mention it in that topic, but maybe parathyroid hormone is released and that increases blood calcium levels. Once those levels get too high again, the entire process is going to repeat. Calcitonin is going to be released. It's going to lower blood calcium levels. It's going to act upon the thyroid again, and calcitonin secretion is going to be decreased again. And exactly what I just said, basically, is what's being displayed in this graphic to the left. Okay, we talked about tropic hormones a little bit before, specifically the releasing and inhibiting factors that are released from the hypothalamus, that are produced from the hypothalamus. But it's important that you understand them and understand that tropic hormones are sort of a separate class of hormones and they have a very important job. So this is one of the reasons why the endocrine system is so finely tuned and it's so finely regulated. Because if it weren't, we wouldn't be able to maintain homeostasis like we do. The tropic hormones are those that regulate the release of other hormones. And there can be multiple tropic hormones released in a singular pathway. And there often are, especially when we're talking about the HPA axis. The first 2 hormones that released are going to be tropic hormones. So the hypothalamus will release CRH, corticotrophin releasing hormone. And that travels to the anterior pituitary lobe via the hypophyseal portal system that we spoke about. The pituitary gland, the anterior lobe, in response to the CRH, is going to release ACTH, which again is another tropic hormone, because it's not really affecting a change directly. It's acting on another endocrine gland to release that specific hormone. So it's indirectly the cause of whatever action it's going to, promote downstream. The ACTH will act on the adrenal glands, and it's going to stimulate the promotion of cortisol, like we spoke about a little bit before. So cortisol is the final hormone, and it's not a tropic hormone. It's the goal. So that's the one that we want. So both CRH and ACTH are tropic hormones because they regulate the release of another hormone. Yes, TSH would be another example of a tropic hormone, yes, because it's being released by the anterior pituitary lobe, and it's going to the thyroid, but it's not doing anything other than stimulating the release of another hormone. So we can regulate the release of T3 and T4 through the release of TSH. So that's basically how the pituitary gland regulates it. And, zooming out even more, that's how the hypothalamus is able to regulate it. So yes, correct. All right, and we're going to do another question. Does anybody have any questions about what we just went over, anything about tropic hormones or negative feedback before we do this? All right, so then I will give you guys a minute or two, take a look. Give me 1 second, I'm going to launch the poll for you. All right. Okay, the polls are live, so take a minute or two and we'll regroup after. Okay, I'll give you guys a little bit longer, maybe another 20 seconds. Get your final answers in. Okay, 1 second. Cool. Okay, so 100% of you said D, which is really cool because D is the right answer. So let's go over it. Which of the following structures release tropic hormones? So we have Roman numeral 1 is the anterior pituitary, Roman numeral 2 is the posterior pituitary, 3 is the hypothalamus, and 4 is the adrenal gland. So basically, just because we just talked about it, it's pretty self-explanatory. So great job doing D. 1, the anterior pituitary does release tropic hormones. Just because, you know, flat peg, FLACTH is a tropic hormone, TSH is a tropic hormone. The posterior pituitary only releases 2 hormones that we know of. It's oxytocin and vasopressin. Neither of those are tropic hormones, so we can eliminate C. So we would just have A, B, and D. The hypothalamus, we know it releases CRH. I think it releases GRH, growth releasing or releasing growth hormone. It releases a few different tropic hormones, those releasing factors that we're talking about. So 3 is also correct. So from that, you can cross out A, and you can cross out B as well, and C, and that will leave you with D. Because 4, the adrenal gland also does not release tropic hormones. It releases catecholamines and a few others. So all of you got D correct. Anterior pituitary and hypothalamus are the ones. Does anybody have any questions about that? Maybe anybody that didn't answer, that didn't participate in the poll? Do you guys need any clarification, or is everyone good on that? Awesome. All right. If you guys think of any questions, again, just type them in the chat.
Yeah, I have a question.
Sure. What's up?
So you said that
You said the posterior.
You have a little bit of an echo.
You said the posterior pituitary release the vasopressin and oxytocin. So is vasopressin another name for ADH?
It is. Yep. So ADH stands for antidiuretic hormone, but it's also known as arginine vasopressin. So sometimes you'll see it as ADH, sometimes you'll see it as AVP, sometimes you'll just see it as vasopressin.
All right, so both of them. All right, just looking.
Okay. Okay, and I put this here just for like a reference, so you guys can look back and maybe study if you want to make flashcards. I think these are pretty good things to make flashcards for. You can use Anki. Just to really quickly go through it, the pineal gland, we're not going to talk too much about it, but it does make melatonin. There are many target organs, and it regulates your biological clock, your circadian rhythm, your sleep-wake cycle. The pituitary gland, it lists some, not all of the hormones over here as well. Gives you some of the target organs and the functions. Thyroid gland, again, doesn't list all of them, but T4 is over here. Target organ function, the adrenal glands we talked about a little bit. Adrenaline and cortisol, there are a few others, but these are the main 2 that we talk about. The pancreas, insulin, glucagon, we said it also secretes somatostatin, but again, these are the 2 main ones. The ovaries, estrogen and progesterone, and the testes, testosterone. So you can look back on this in your free time if you want to use it. It's a pretty good resource if you want to make some flashcards or anything like that. And then we're just going to do a quick review of the individual hormones that we spoke about, just talking about where they come from and what their function is in the body. So follicle stimulating hormone, a lot of these we have already touched upon in other lectures, so I didn't want to go too in detail. I just wanted to put a quick little blurb here for you to look back at if you just need like quick clarification on what something does if you forget. So follicle stimulating hormone is released by the anterior pituitary. It encourages the production of ova in females and the production of sperm in males. Luteinizing hormone is released also in the anterior. It's responsible for the release of the egg from the follicle in females and the production of testosterone in males. Remember, testosterone comes from the cells of Leydig that are in the testes in males, and that is promoted by luteinizing hormone. So adrenocorticotropin hormone, ACTH, is released by the pituitary, the anterior pituitary gland. and it acts on the adrenal cortex to stimulate cortisol secretion. So that is one of those tropic hormones that we just talked about. Thyroid stimulating hormone is also one of those tropic hormones, and it is released by the anterior pituitary, and it acts on the thyroid to secrete T3 and T4. Prolactin, which we spoke about, I think I spoke about that a little bit in my development lecture. It's also released by the anterior pituitary. It's not a tropic hormone because it actually directly affects the target tissue. So it promotes milk letdown from the mammary glands during breastfeeding. So it allows milk production. And there's like a surge of prolactin every time a mother goes to breastfeed. And as long as the mother is breastfeeding, the anterior pituitary will produce prolactin. It also releases endorphins. I think specifically it's beta endorphin. So the anterior pituitary releases these endorphins, and they act on opioid receptors in the body. So they create a sense of calm, and they reduce perception of pain. So those are sort of like the natural molecules that act on the natural opioid receptors in your body. And they're produced by the anterior pituitary. So growth hormone, sometimes it's abbreviated as GH, and it is released by the anterior pituitary as well into the bloodstream. And it just stimulates growth and a lot of pro-proliferative cell functions throughout the body. So it affects a lot of different tissues. Insulin and glucagon are produced by the islets of Langerhans, like we spoke about before, in the pancreas. So they decrease and increase blood glucose respectively. So insulin is going to decrease that blood glucose by letting the surrounding tissue uptake glucose, and glucagon is going to increase it by mobilizing those glucose stores. So glycogenesis, gluconeogenesis, it's going to get glucose into the blood. And T3 and T4 are both produced by the thyroid, and they circulate the body to increase basal metabolic rate. Calcitonin is also produced by the thyroid. It decreases the calcium level in the blood, like we spoke about a little bit before. And parathyroid hormone is secreted by the parathyroid gland, and it opposes calcitonin by increasing blood calcium levels. Okay, so not too in depth, but just a quick review of those hormones that I wanted. And we can talk a little bit about the digestive hormones. Like I said, we do have a digestive lecture that's going on next week, but the hormones in the digestive system do come up a lot because it is ruled. It does interact with the endocrine system pretty frequently in order to function properly. So I have these three and then another two, I believe, on the next slide. So cholecystokinin is usually abbreviated as CCK. These are the three most important ones that I usually see in terms of endocrine functioning of the digestive system. CCK is released by the duodenal epithelial cells when food enters the stomach. It stimulates the contraction of the gallbladder and inhibits gastric emptying. So it sort of constricts the pyloric sphincter is the structure that is right at the end of the stomach. It connects the stomach to the first part of the duodenum. That's called the pyloric sphincter. So the cholecystokinin actually inhibits gastric emptying by contracting that pyloric sphincter. And that's usually, it's released when food enters the stomach, and it's inhibited with acidic conditions entering the duodenum. So acidic conditions in the duodenum would mean that the bolus, which is the term for food after it's being digested and mixed in with digestive juices in the stomach, when that moves to the duodenum, it creates an acidic environment, and CCK is released, and it slows gastric emptying. Because the food is moving along properly. So we don't want too much food to enter the duodenum before it's properly digested. So CCK is released, and it inhibits gastric emptying. Gastrin is secreted by G cells in the cell wall of the stomach. It stimulates acid and pepsin secretion and gastric motility. So it actually increases gastric motility. So gastrin and CCK sort of work together to make sure that there's proper flow of gastric emptying into the duodenum. And gastrin is also stimulated when food enters the stomach. And then the third one is secretin. Secretin stimulates the flow of bile to the gallbladder and stimulates the release of bicarbonate from the pancreas. So CCK stimulates the contraction of the gallbladder to actually release the bile through the common bile duct into the duodenum. The secretin is actually what allows the bile to go from the liver to the gallbladder. And it stimulates the release of bicarbonate from the pancreas, and bicarbonate release is a response to the acidic bolus reaching the duodenum, kind of like CCK. So your stomach is pretty acidic, and it needs to be acidic in order to denature proteins and start chemical digestion the way it's supposed to be started. But once it enters the duodenum, it actually needs more of an alkaline environment, more of a neutral environment. And I'm getting a little bit out of the scope of this specific lecture, so we'll definitely talk more about this during the digestive lecture. But secretin is responsible for the bicarbonate release into the duodenum, and it is because of that acidic condition that reaches the duodenum. Okay, and two other digestive hormones that I see a lot on the MCAT are ghrelin and leptin. So ghrelin is the hunger hormone. It is released by the stomach that stimulates the appetite and promotes fat storage. So ghrelin levels peak before meals and during fasting. So that really is what stimulates your appetite. It drives us to eat. If you ever experience hunger pangs, sometimes it feels like your stomach is kind of squeezing up. That is pretty much directly a result of a spike in ghrelin levels before you're supposed to eat. So it really promotes the behavior of ingesting food. And ghrelin and leptin sort of work together.Ghrelin's the hunger hormone. Leptin is the satiety hormone. It makes you feel full. It promotes feelings of fullness and satiety. It is released by fat cells, and it is received by the hypothalamus. So that's also sort of a negative feedback mechanism. The more food you eat, the more fat cells are going to be present, and the more fat cells there are, the more leptin is supposed to be released. So that should, in theory, suppress your hunger until the fat cells, they're used up for energy. But we see leptin resistance. It's thought to be the main biological factor that contributes to obesity. So when you have high levels of leptin, you would expect to see someone who is very full and someone who doesn't want to eat. But when we look at the morbidly obese population, we actually see that leptin levels are extremely high compared to the average population. So one of the theories is that these individuals are leptin resistant. So they have a lot of leptin, but for whatever reason, the receptors on the target tissue, they are not as effectively accepting leptin as like the average population. So that contributes to obesity because there's no feeling of satiety or fullness. So the person will just eat. And that's why bariatric surgery comes into play. That's why it's so effective when compared to dieting, because long-term, when you're on a diet, your ghrelin levels will spike, and they'll maintain a very high level, even, for months after you start dieting. So it's very hard for a person to change their body's set point. They're like our homeostatic compensatory mechanisms that include ghrelin and leptin that keep, morbidly obese people morbidly obese. So it's a lot more than just psychological. It's psychological, biological, a bunch of different things. So that's why bariatric surgery also, it alters the anatomy of the human, but it also alters the physiology of endocrine functioning. So there are a couple of different procedures. I actually work at a bariatric surgery practice, so if anybody's interested, I'm definitely open to answering some questions. But a lot of the main surgeries reduce the size of your stomach, so it restricts the amount of food that comes into your stomach. And as we spoke about, your stomach is also a secretory organ. So it releases ghrelin. So when we remove part of the stomach, we're actually removing the parts of your body that are secreting this hunger hormone. So it helps in that aspect as well. And I have seen a couple of things on the MCAT regarding bariatric surgery and ghrelin and leptin levels and treatments for the morbidly obese population. So these are definitely some important things to know. Does anybody have any questions about that before we do this one last question? This is pretty much it. Okay. I'm going to relaunch the poll. Take a minute. Shouldn't be too hard of a question, but we'll regroup and discuss in a little bit. All right, take another few seconds and I'm going to close the poll and we can go over it. Okay. Interesting. Okay, so a third of you said A, a third of you said C, and a third of you said D. So the correct answer is A, blood. If you didn't specifically know like what gastrin is or how it's released, a good jumping point is that it is a hormone. It's an endocrine hormone. And by definition, endocrine hormones are released into the blood and they circulate throughout the body. So the extracellular matrix, would probably be, no, that probably wouldn't even be the second best answer. It just wouldn't be released into the extracellular matrix unless like they would specifically say like some protein is being used for like juxtacrine signaling or paracrine signaling, maybe. But in terms of like regular hormones like gastrin, I don't think that that would be a feasible response. Digestive duct. I don't know if there's like a specific structure called the digestive duct. I just put digestive generally. If any sort of like hormone molecule is being released into a duct, it's considered exocrine, sort of like those enzymes that we'll talk about during the digestive system lecture. They're not really considered true hormones. Gastrin is a true hormone, so it's not released into a duct. And likewise, it's also not released into the lumen of the intestines. It's produced by the stomach, and it actually it is released directly into the bloodstream. So all three of these, they're secreted by structures that are part of the digestive system. So CCK and secretin, I believe, CCK is released by the duodenum, but it's not released into the lumen of the actual duodenum. It's released into the blood, and that's how it travels and affects the target cells. Same thing with gastrin. It is released by the stomach, and it is released into the blood. And same thing with secretin. It is released, I believe, I think it's also released by the duodenum, but it goes into the blood and then it affects target cells by traveling through the circulatory system. And that's all I have for today. So our lecture tomorrow night, I believe Janelle is teaching sociology from a demographic and macro lens, and it will start at 8:30 p.m. sharp. And then on Sunday, we have our office hours. So if you have any questions about David's lecture last night, my lecture tonight, or Janelle's, you are more than welcome to stop by and ask some questions. But other than that, we should be all finished. And micro lens, she's going to be teaching. Cool. So that's all of the content for tonight. So if you have no questions, have a great night. If you do have some questions, I'll stick around for a few minutes so you can unmute or type them in the chat box, and I will do my best to answer them. But like I said, otherwise, have a great night.